Alright, so now let's talk about translation. This is when our messenger RNA is converted to create an amino acid which creates our protein. So translation, you could think of it as translating that mRNA to that protein. Now there are three different structures that constitute for the process of translation. The first one obviously is our messenger RNA, which as I talked about will be converted to create a specific protein. Now the second one is known as our transfer RNA or tRNA, which is a small RNA that binds the amino acids from the cytosol and helps code for the specific RNA. It takes that specific amino acid to code for a specific anticodon in our ribosome, which we'll talk about later. To memorize the tRNA in a simple way, you could think about it as just a small RNA that binds an amino acid. So what does a tRNA look like? So here's what a 2D version of the tRNA looks like. We'll see a 3D version later. So we have our attachment of protein or our site of protein in which protein attaches. Now our second structure is known as our anticodon. Now our anticodon will attach to a specific part of the codon from the mRNA. So let's just say we have adenine, uracil, and guanine. Well, the anticodon over here would be uracil, adenine, and cytosine. So that's what an anticodon is. You can think of it as the attachment site. Alright, so now let's talk about our third and final structure responsible for translation, and that includes the ribosome. Now the ribosome is just a small reading machine found in the cytosol. It's the one that codes throughout the whole mRNA and makes that specific protein. So there are two subunits of an active ribosome in translation. First we have our small unit and we have our large unit. Now both of these fuse together in translation to code for our RNA. Now in our ribosome we have three different binding sites for our transfer RNA. The first one includes our A site. Now the A site will be the first site in which our tRNA will attach to. The second one is called the P site, and the P site is the second site in which the tRNA binds to. Now the third binding site is known as our E site. The E site, as I talked about, is the final binding site on the ribosome. So this is how it goes in order. The tRNA will go through the A site, then the P site, then the E site. We'll see this later. Alright, so what does translation look like in action? So we're first going to talk about the first process of translation. This is called initiation. This is when our mRNA is already produced and it goes through the pores of the nucleus. When it does, it coils around and makes this sort of loop in which our subunits are different subunits. We have our large subunit right here and our small subunit right here. They bind together to form an active ribosome, which goes around the mRNA, just reading all throughout the mRNA and all the codes. It first goes to the start codon, which is usually AUG. So now we have a second stage of translation, or the second process, and that is known as elongation. So we zoom into the ribosome, and this is what we see. Now this starts by the initiator tRNA, and unfortunately this picture does not show the initiator tRNA. So I may be wondering, what is the initiator tRNA? Well, this is basically the start tRNA, and it usually adheres to the P site where the whole ribosome attaches to the start codon, the AUG. When that does happen, our tRNA, our initiator tRNA, goes to the P site and carries methionine, or also known as MET. So let's just say this is our tRNA, this is our initiator tRNA. It's really not, but let's just say it is, and it attaches to the P site. Well, that's great. Now we have the tRNA, and this is just a random tRNA, and it'll go through the pool of amino acids. Now, as we talked about through the anatomy, they have a site in which they attach the protein, but they sacrifice an ATP molecule, and they take that, they go through to the A site. Now when this does happen, the new tRNA will replace our initiator RNA, our initiator tRNA, and the initiator tRNA will move to the E site, in which they just leave. They're pushed out. So here's what our new tRNA looks like. It has a methionine and another amino acid that is specific for this specific codon. So that process continues. A new tRNA gets another amino acid and just goes to the A site, pushes that old tRNA that we talked about, and that old tRNA goes to the E site, and that process continues and continues until there is a buildup of amino acids which form the strand of protein, as we all know. Now we're at our third stage of translation. This is known as termination, and this is when our ribosome contacts a stop codon. When this does happen, there's a release factor that adheres to the A site instead of another tRNA. When this happens, the protein is taken off of that ribosome, and the ribosome usually splits off. But since those two subunits are close to the RNA, they just bind together and they go through this whole process again. So now we have come upon our fourth and final stage of translation. This is known as packaging and exportation of protein, and this is when our free protein docks the rough endoplasmic reticulum. When it does, there are enzymes that fold this whole protein to fit in a transport vesicle, which is taken out to the Golgi complex, and the Golgi complex usually puts lipids in there and then sends it out of the cell.